Our next presenter is Sri Krishna. Sri is from Arizona State University in the Biological Design program. His major professor is Dr. Karen Anderson, and the title of Sri's presentation is T-Cell Antigen Discovery Strategies for Viral and Cancer Immune Therapies. Here we go.
Sorry, my first slide is not here, by the way. Okay. All right. So the human papillomavirus, or HPV as it's commonly called, caused about 500,000 cases of cervical cancer worldwide last year. It's also been associated with an increasing number of other different types of cancer, such as head and neck cancer, anal, vulvar, and penile cancer. My research project is about using your own immune system to fight against HPV-associated cancers. Now, I know what you're thinking. There's already a vaccine out there for HPV. But if all of us were vaccinated against the virus, I wouldn't be standing here talking to you about my dissertation project. Less than 50% of the population in the US is even vaccinated against HPV. And the vaccine does not protect you after you've been exposed to the virus. Now, most of us know that we have an immune system in our body which constantly fights and prevents these infections. Much like a constellation made of several different types of stars, the immune system is made of several different types of immune cells which constantly interact with each other, communicate with each other to make an immune response. One particularly important type of immune cell that I study is called a T cell. If you have an infection, there are two ways to eliminate that infection. One is to prevent that virus from even entering a cell. That's what antibodies do, and that's what's in your HPV vaccine. But what if the virus has already made its way into the cell? What if it's already established an infection? Your HPV vaccine or antibodies isn't going to do anything then. That's where T cells come into play. T cells, I like to think that they work like CIA operatives, where they go from cell to cell looking for signs and clues of infection. Oftentimes, these clues of infection come in the form of protein bits from this virus itself. So when a virus infects a cell, some of these protein bits get stuck on the cell surface, and the T cell can see it. When the T cell sees these infected cells, it determines that the cell has to be destroyed, and it does it really effectively. So this is a T cell trying to kill a target cell here, which is a tumor. Now you can imagine, if you know all these protein bits from a given virus, you can start thinking about making your own vaccine or treatment using T cells. And that is exactly what I do in my dissertation project. We've already discovered several of these HPV protein peptides, which are parts of the virus itself, so that we can train these killer T cells and make it attack the target cell. My end vision is that if a patient walks into the clinic with HPV-associated cancer, you can take out their own killer T cells from their own blood, train it outside with these protein bits from the virus, and put it back into their own body so that their own T cells can fight against their own tumors, making it an effective, personalized immune therapy. Thank you so much for your attention and this opportunity.